Hello, this is Dr. Hanaa Assil, and this is about diffusion and osmosis. So let us start by talking about diffusion. What is diffusion? Diffusion is the net movement of molecules from a region of their higher concentration to a region of their lower concentration down the concentration gradient, and this is a result of random movement. So what does that mean? If I have a crystal of potassium permanganate at the bottom of a beaker, so that area at the bottom of the beaker has a lot of potassium permanganate, so that is area of high concentration. But at the top of the water, there's not that much potassium permanganate, so that is area of low concentration. So the particles of potassium permanganate will move from where there is lots, so that's area of high concentration, to where there is less, and that's area of lower concentration, until they are equally distributed in the water, so that all the water takes that purple color of potassium permanganate. Now, that means that where I have lots, that's area of high concentration. Where I have less, that's area of low concentration. And this gradual change in concentration from high to low, this is what we call concentration gradient. So the particles move down their concentration gradient. So where do we have diffusion in living organisms? If we're talking about a leaf, you know that a leaf has holes mainly at the bottom, which we call stoma, or all of them are called stomata. Now, there are gases that diffuse in and out from the stoma. So carbon dioxide, for example, has to go in to be used for photosynthesis. Oxygen can go in or out depending on whether the plant is doing photosynthesis. So all of these move by diffusion. Something else that goes out of the leaf by diffusion is water vapor. If you remember, we have something called transpiration, and during that process, water vapor moves out of the leaf. All of these move by the process of diffusion. What about in our body or in animals in general? For example, in the lungs, we have diffusion of gases between the alveoli and the red blood cells in the blood capillaries. So carbon dioxide moves out of the blood into the alveoli, so this is by diffusion, and oxygen moves from the alveoli to the red blood cells, and that is also by diffusion. So let's take a look at this question. This question says, which substances may diffuse in and out of plant cells? So into the plant cell, what may diffuse into the plant cell? You should be able to see that out of these options, oxygen can diffuse into the plant cell. Water can diffuse out. Remember, we said by transpiration. So our answer here is B. Okay, another example of diffusion is if I have ammonia, concentrated ammonia solution on a cotton wool on one end of a tube, and I put moist litmus paper at different distances along the tube. Now, if I ask you, is ammonia an acid or a base? Is it acidic or alkaline? You should know that ammonia is alkaline, or it's a base. So if I put ammonia on one end, it should diffuse from area of high concentration to area of low concentration. So how do I show that? I can put red litmus paper so that as ammonia diffuses from area of high concentration to area of low concentration, these litmus papers will start to change color. Ammonia is alkaline, so as it moves, the red litmus paper becomes blue, and then the next one becomes blue, and then the next one becomes blue, and so on. So this indicates that it is moving from where there is lots to where there is less. Another process is called osmosis. And what is osmosis? It is the net movement of what? Of water molecules from a region of higher water potential. The word higher water potential means where we have more water. So when we have more water, that means we have a dilute solution of a salt or sugar solution. If it is dilute, it has a lot of water, so we say it has higher water potential. So this is, first of all, movement of water molecules from higher water potential to lower water potential. Lower water potential means there is less water, and that means the solution is more concentrated. So I have a concentrated salt solution, for example. And this has to be across a partially permeable membrane or through a semi-permeable membrane. So in osmosis, we have a side where we have lots of water, high water potential, that's a dilute solution. We have another on the other side, which is low water potential, which is a concentrated solution. And these two solutions are separated by a membrane. And this membrane has to be partially permeable or semi-permeable. That means it's a membrane that allows only some molecules to pass through, but not others. Usually it will allow only small molecules, not large molecules. So, if we say, first of all, which row describes the movement of water by osmosis? We said, what is osmosis? It's from a region of high water potential to a region of low water potential through a partially permeable membrane. So please remember that this is definition of osmosis. So another illustration of osmosis. If I have a thistle funnel, and you should know that a thistle funnel, now this is a thistle funnel that is inverted. It has, at the bottom, we closed it with a semi-permeable membrane. Now this semi-permeable membrane is a sort of cellophane bag or a sort of whisking tubing. These are examples of things that are semi-permeable. And I fill, or I put inside the thistle funnel, I put sugar solution. Outside I have water. So outside I have higher water potential. Inside I have a sugar solution, and that is a lower water potential. Can you see the initial level of solution in the thistle funnel? What if I leave this for a while, for some minutes, 30 minutes, an hour? I will find that the water will move in from where there is lots of water outside to where there is less water or lower water potential inside the thistle funnel. Can you see what happened to the level of the liquid on top? Of course, the level of the liquid rises. So this is an illustration of osmosis. Another one is where I have something called a whisking tubing. So remember, if he mentions a whisking tubing or a cellophane bag or a dialysis tubing, all of these are kinds of semi-permeable membranes. That means they will allow the water to move in and out, but they will prevent large molecules from moving in and out. So if I have, again, outside I have water, inside I have a sugar solution, how will the water move? Remember, it will move from where there is higher water potential, so in this case it's outside, and it will move into the whisking tubing because there is a more concentrated solution inside the tubing, and that means that has lower water potential. Again, if he says he has a dialysis membrane, again, a dialysis membrane is a kind of semi-permeable membrane. Now, what do I have? Inside this dialysis membrane, I have two types of molecules. I have starch and sugar. And you should know that starch are large molecules. sugar or glucose or maltose, these are smaller molecules. So if I put this into my dialysis membrane and there is water outside, now if I'm saying that at the beginning inside the tube I have starch and sugar, now if I leave it for some time, the sugar molecules go out of the tubing into the beaker. Is this diffusion or osmosis? If we're talking about movement of the sugar molecules out of the tubing, this is diffusion, not osmosis. Osmosis is when we say the water moved into the tubing so that the tubing became larger, for example, or swelled up. Then in that case, that is osmosis. But if I'm talking about movement of the sugar molecules out of the tube, because the sugar molecules are small, the starch molecules are large, so they will not be able to pass across the semi-permeable membrane. But movement of the sugar molecules out of the tube, that is diffusion, not osmosis. Okay, what happens in animal cells? Remember, an animal cell has a cell membrane. And you should know that a cell membrane is a partially permeable membrane. So if we have a group of cells, these are animal cells, they have only cell membrane surrounding them. Now, if there is water, more water potential in one of these cells, then the water will move from cell to cell by osmosis. This is movement of water from one cell to the next because one of them has higher water potential than the other. This is by osmosis across the semi-permeable cell membrane. What about in plant cells? You should realize that when we have root hair cells or any plant cells, they have two things. They have cell wall and cell membrane. The cell wall is fully permeable. The cell wall will allow anything to pass through. But then they come to the cell membrane. The cell membrane is partially permeable. So if we want to take up water from the soil, then the water will have to go from the soil. If there is higher water potential in the soil, then the water will move from the soil into the root hair cells across the semi-permeable cell membrane. This is by osmosis. And then we move from one cell to the next, again across the semi-permeable cell membrane by osmosis. So if I have plants in a solution, well, one of them wilted and the other one is not wilted. What happened? The one that wilted must be in salt solution. Salt solution means there is higher water potential inside the root hair cell, so the water, instead of going in, it went out. If we have salt solution outside, then outside there is lower water potential, so the water will move from the root outside. And that is why the plant wilts. While the other plant that is in water, well, there is higher water potential outside, so the water went into the root by osmosis, and that is why the plant has enough water. Okay, so we need to talk about the effect on cells of putting them in different water potentials. So if I have an animal cell, so this is an example of an animal cell, like red blood cell. Now I put it in what? I put it in concentrated solution. So what is surrounding it has very low water potential. Then what happens? If the water potential inside the cell is higher than outside, that means I have more water inside than outside, then the water moves out of the cell by osmosis. This causes the animal cell or the red blood cell or the liver cell to shrink. So that is the effect of putting an animal cell into concentrated solution or a solution with low water potential. What if I put my red blood cell in water or in a dilute solution? Now in that case, If outside is dilute solution, lots of water, then the water potential inside the cell is lower than outside, so the water will move into the cell by osmosis, and the cell will start to burst. Now remember, this is an animal cell, a red blood cell or a liver cell, and it has only a cell membrane surrounding it. If it starts to swell, eventually it will burst. So this is an animal cell in higher water potential, or in distilled water, or in a dilute solution. What if I put my red blood cell into a solution in which the water potential inside the cell and outside are equal? If the water potential outside the cell is equal to the water potential inside the cell, then there will be movement of water molecules, but the amount of water going in is equal to the amount of water going out, so we say there is no net movement of water molecules. Are we paying attention? So I put my red blood cell into a solution that has the same water potential as that inside the cell. So inside and outside are equal water potential. What happens? Please do not say no water will move. That's not right. The water will move, but we say there is no net movement, because the amount going in will be equal to the amount going out. So there is no net movement of water molecules. What if we have a plant cell? You know that a plant cell have a cell wall and a cell membrane. Now I'm going to put it into what? Into a solution that is concentrated solution. That means it has low water potential. What will happen to my plant cell? Well, if I put it in a concentrated solution, concentrated salt solution, so this has low water potential, and that means the water potential inside the cell is higher than outside. So what will happen to the water? The water will move out of the cell by osmosis. Now, when the water moves out of the plant cell, we said animal cell, if water moves out, the cell just shrinks. Now, when the water moves out of a plant cell by osmosis, that means the cell wall will shrink a little bit, the subvacuole will shrink and become very small, the cell membrane will shrink away from the cell wall, and we say, can you see this? The cell membrane originally was right next to the cell wall. Now everything is shrinking in, so the cell membrane shrinks away from the cell wall, and we say this cell has become plasmolysed, or we say that it has become flaccid or wilted. So if I put a plant cell, now plant cells like what? In the lab we use, for example, potato cells. So if I put a piece of potato in concentrated salt solution, then the water potential inside the cell is higher, water moves out, the cell shrinks, the cell membrane shrinks away from the cell wall, and we say the cell has become plasmolysed. What if I take my plant cell and I put it in high water potential? That means I put it, for example, in a beaker containing distilled water. So I'm going to take my piece of potato, put it in water. What happens? In that case, there is water potential inside the cell lower than outside. And that means water will move into the cell by osmosis. Now, when water moves into the cell, then everything will swell, the subvacuole becomes larger, the cell membrane is forced against the cell wall. Now, my problem is the cell wall is rigid, it will not expand anymore, and that means my cell becomes turgid. Are we following? When we were talking about animal cell, we said the animal cell, if water goes into it, it will continue to swell until it bursts. But the plant cell has cell wall. So the cell membrane will be forced against the cell wall, and the subvacuole will have a maximum volume. It cannot become bigger than that. And we say the cell has become turgid. So let's take a look at this question. The diagram shows an experiment using an uncooked potato. The skin of the potato was removed as shown. Which diagram shows the results of the experiment after 24 hours? Well, let's see. Outside the potato, we have water. Inside the potato, we have a concentrated sugar solution. So what will happen? The water will move from outside into the potato. So the water outside will become less and the solution inside the potato will become more. So my answer is C. An animal cell placed in a solution with water potential lower than its cytoplasm. Remember, lower water potential means more concentrated solution. So I'm putting my animal cell into a concentrated solution, concentrated salt solution, for example. Which statement correctly describes the movement of water across the cell membrane? So if I put it into water potential lower than what is inside, how will the water move? If inside has higher water potential, then the water will move out of the cell. We have net movement of water out of the cell. A cytoplasm of a plant cell contains 15% sugar solution. The plant cell is placed in sugar solutions of different concentrations. In which solution would there be a net diffusion of water out of the cell? So what do we have? We said the plant has 15% sugar solution. Now I want the water to go out. For the water to go out, then outside should be lower water potential, and that means a more concentrated solution. So for the water to go out of my plant cell, it must be going to something that is more concentrated than what is inside. So that means something that is, for example, 20% sugar solution. If it is 20% sugar solution, that means it's a more concentrated solution than the one inside the plant cell. So water will move out of the plant cell. Okay, the diagrams show 4 similar animal cells immersed in blood plasma. The black dots represent molecules of dissolved oxygen. Which cell will have oxygen molecules diffusing into it most rapidly? Where do we have faster diffusion? If we have lots of oxygen outside, then more of it will diffuse in. So which one has more concentration of oxygen outside? Well, that is C. If I have lots of oxygen outside, then more of it will diffuse into the cell. What about this one? He says 2 pieces of potato are cut to have exactly the same mass and shape. The mass is measured and recorded. One piece of potato is placed in water and the other piece is placed in concentrated salt solution. They are both left for 1 hour. The mass of each piece of potato is then measured again. What happens to the mass of each piece of potato? Now, what happens to the mass of each piece of potato? We have the first potato in water. Remember, if I have lots of water outside, then the water will go into the potato. So if I weigh it after that, the mass will be increased. What if I put the potato in concentrated salt solution? Well, that means I have lower water potential outside. There's lots of water inside the potato, so the water will move out of the potato and the mass will decrease. Are we following all of this? What about this question? Which arrow represents osmosis? This is a plant cell, and I put it in concentrated salt solution. If I put this plant cell in concentrated salt solution, first of all, will the water go in or out? Well, concentrated salt solution outside means there is higher water potential inside the cell. So I'm looking at an arrow that goes out of the cell. So are we talking about C or D? Remember that the water will move out of the cytoplasm of the cell. This cell is how we draw a plasmolysed cell. So the cell membrane has shrunk away from the cell wall. So C is actually a space between the cell membrane and the cell wall. But he's asking what is the movement of the water. The water will move out of the cytoplasm of the cell. So my answer is D. What about this question? The diagram shows 3 plant cells. He has cell P, cell Q, cell R. And he's giving me the water potentials of each one, and he's saying what is the correct net movement of water by osmosis. We said, how does water move by osmosis? From area of high water potential to area of lower water potential. So for each of these cells, the water will move from higher to lower. So how does water move? Can you see that it will move this way? And that means my answer is A. Okay, the last question here says, a student cuts a potato into cylinders as shown. The student states that when the potato cylinders are put into salt solution, the concentration of salt solution will affect the length of the potato, but not the mass. Plan an investigation to test whether this statement is correct. So this is a question from paper 6. You're required to plan an investigation to test whether the statement is correct. In your plan, include the apparatus needed, including a label diagram if you wish, a brief description of the method, including how you will treat variables, and any safety precautions, the measurements you will make, and how you will process your results to draw a conclusion. So this is usually a question in paper 6 that has about 7 marks. So let us see what we should do. So this is what we're required to do. We want to prove that the potato cylinders will be affected by the concentration of the salt solution will affect the length, but not the mass. And he wants these list of things. Make sure that at the end of your description, you have included answers to all of this. So let's see. How do we do this? First of all, I'm going to tell him, cut equal sizes of potato cylinders from the same potato. So these are the things that we're keeping the same. Weigh the mass of each one using balance. We have to mention apparatus. Prepare salt solutions of different concentrations. You should actually have more than 3 or 4 or 5 concentrations. Don't say 1 or 2. Place equal volumes into different test tubes. Put potato cylinders into each test tube. Leave at room temperature. So you're telling him that the temperature is the same for 2 hours. So that means the time is the same. Can you see the variables that you're keeping constant? After that, what should we do? Remove each potato cylinder, measure its length using a ruler. He says it will affect the length but not the mass. So I need to measure the length and measure the mass. So measure the length using a ruler, dry in an oven, remove the dry potato, and then here you have to mention something about the safety. You put it in an oven, you want to remove the dry potato using gloves to avoid burning the skin. Okay, what are we going to measure? We're going to measure the mass of the potato using balance. And then you're going to draw a table of initial and final lengths. So this is how you're going to treat the measurements. Draw a table of initial and final lengths and masses of each potato at different concentrations. Determine the difference in length of the potato and determine that the mass has not changed. So in this case, you have given him everything that he is looking for. Okay, so that's the end of this video. I hope that this was useful to you, and please continue listening to the videos, and thank you.